All right, hello everyone. Thank you for coming and listening to me today. My name is John Malloy. I work with Sarah Walker at Arizona State University. I'm in the School of the Earth and Space Exploration, and I'm a PhD student there. I'm here to talk about how metabolism and genetics are not useful for distinguishing life on Earth, and are even less useful for predicting what life could look like elsewhere. The main motivation behind this is kind of going against researchers like Stephen Jay Gould and other evolutionary biologists like him. Their idea of life is that if you were to start over from scratch, if you were to go back 4 billion years and say life started again in a different place, it would look fundamentally different than what we have today. Essentially, evolution is random. I think he's wrong. And I think that kind of view of evolution is wrong as well. I would think that if life restarted, maybe you won't have humans, maybe you won't have humanoid bipedal organisms, but you would end up with similar properties to what we see today. I think of chemistry as, and life, as a universal property. Your life has universal properties that can allow for prediction, allow for emergence. I think of things as a network scientist at times, so network properties may come in here a little bit, but the idea ends up being there are certain properties of life that will allow for kind of prediction of what can happen in the future. And that's going to be my argument that I'm going to make today. Kind of starting off kind of very, very basics. I think then I have to define what life is or define how I view life. Starting off with that, it's a biosphere level property. Life can't be looked at a single organism level. We are not the only things that are alive for sure, as well as it can't be an environmental level. Everything in the ocean does not consist of all of life. Life is something that is general to the entire planet. It started in one place and has since evolved and radiated out from there. And also, life is a chemical-based property. Have the TCA cycle up here. It's the way that we as aerobic organisms get our energy. Other organisms use slightly different ways of doing this. But the idea is that we use chemistry to keep ourselves going. And everything on Earth, every living thing on Earth, is fundamentally based on this universal chemical language. There is nothing on Earth that does not use chemistry at its core. And ultimately, you can look at life and look at the chemical properties of it and kind of describe it in a chemical sense. So in reality, I say kind of I'm a network theorist. I'm really a chemist trying to describe life as a chemical property. And beyond just life, these kind of chemical properties can be universal and can be quantified. So not only can they describe life on Earth, but life elsewhere also would have some kind of chemistry element to it. There is nothing in the universe that is not inherently based in chemistry. And we will be able to quantify and compare how our life on Earth looks chemically to something else. So it gives us a lot of flexibility in what this can do. So what kind of properties am I talking about? How can we actually compare these two? One kind of specific thing is a chiral property. Chirality, just as a quick primer for that, is a property of molecules where they can be either asymmetric or symmetric. So one of the ways, if you take your hands, put them out in front of each other, if you try to put them over each other, kind of superimpose them, you're not able to do that. Your thumb and pinky will never be able to line up. They'll never be able to block each other. Since you cannot be superimposed, your hands are asymmetrical. Therefore, they are chiral. This is kind of a definition thing. In achiral objects, like a water bottle, you can put a left-handed water bottle, right-handed water bottle. That doesn't even mean anything. They can be put in front of each other. It's the same object, it's symmetrical, therefore it is achiral. And this is an interesting property for life because a lot of the molecules we see on life have one or the other chiral property. For example, all the amino acids that we see, that we use as part of our biochemistry, they're all left-handed amino acids. There is no right-handed amino acid that we can use in our biochemistry. And that's consistent across all of life. Sugars are right-handed, amino acids are left-handed. It depends on what kind of thing you're talking about. But this is a property that persists across life as we know it. There are other kind of properties we can use to compare as well. You can look at molecular weight. Literally, how much do the compounds in our body weigh? Compounds in our biochemistry specifically. Look at elemental distributions. What kind of elements are we using? And then you can start looking at more kind of complex, quote unquote, measures. You can build chemical reaction networks. What kind of compounds turn into other ones, and what's the patterns in how they get transformed. But complexity in quotes, because that's hard to define. But let's say about let's say you define complexity as the number of steps it takes to generate a certain compound. You can start looking at that when it comes to chemical reactions. So there's a lot of properties we can use to kind of distinguish all these different chemical things. And they can be something that we can compare across biochemistry here and potentially to chemistries elsewhere. To give you a little bit of kind of data here, something that we've worked on here at ASU is the kind of patterns of the biosphere in terms of chirality. So kind of break this down for a second. We are able to get all of the compounds that we know of in biochemistry from a database called KEGG. It's a well-used standard database, and it's about 7,500 compounds or so in those. And what we can do here is say, out of all those 7,500 compounds that we see in biochemistry, how many of them are chiral? How many of them have this asymmetric property? And it turns out roughly 64% of them have it. So, okay, cool, that's interesting. Let's get a little deeper. You can start looking at all of these different properties across all these different organisms. Look at the Joint Genome Institute, for example, has a number of amazing data sources on this. They're able to look at the 3 domains of life, bacteria, archaea, and eukarya, and say for all the bacterial compounds which they have sequenced and categorize the compounds inside those organisms, what's the chiral pattern across all of those? Interestingly, for bacteria and archaea, actually for everything, as the number of compounds increase, the chirality of the organisms decrease. So as these organisms get larger, they have less chiral compounds. That's interesting, right? That's a chiral pattern. Why does that pattern exist? If you look at something where you have a metagenome, which is where you go to a particular environment, get out all the DNA in that particular environment, sequence all of it with no regard to what kind of organisms you have there, you're going to have a bigger sample because you're going to simply just have more things there. But also, you're going to have a slightly different property. As the number of compounds increase, yeah, the chirality still decreases, but it starts to asymptomatically approach the 64% line that you see in biochemistry as a whole. Why does that happen? Again, there's a lot of interesting patterns to this that, again, we're not entirely sure. But something that we can say from this is say, if you were to find this elsewhere, if you find these similar patterns somewhere else, this may be a signature of life. This may be something that say, this is a pattern that we see in life here on Earth. It could potentially mean life will go toward this pattern elsewhere. So a way I look about this, kind of the final thought I'll end on, is that there are certain ways to explore chemical space, and these properties can predict what elements of chemical spaces will be explored by certain subsets of. So here, x-axis, I'm just saying some kind of complexity measure. Leave it up to your personal view of what complexity means. Again, I'm not going to define it here. But on the y-axis, I have different elements, smaller elements down here, kind of getting into the heavier, more metallic, more radioactive elements as you go further up. And kind of getting a quick example, if you look at things like the geosphere, things that are not alive show up in the Earth. There are all kinds of elements spread all throughout this geosphere. We have all the metals down to hydrogen, but they're not that complex. You're not going to get something that's terribly hard to make or terribly like structurally complex, wherever their definition is, just by looking at the geosphere. You're going to get probably some mercury by itself, for example, so get some gold in the Earth. It's not going to do much with it. Differently, if you look at the technosphere, things that we as humans are able to make in our labs, things that we're able to experimentally produce, we can make all sorts of weird stuff. We're able to put gold into biochemical systems and use it as a tracker for how well organisms grow. We're able to put metal cofactors into drugs. We're able to do all sorts of weird stuff with our chemical knowledge that we have. And I would argue it's pretty complex by whatever measure, again, you're using. But most interestingly, the biosphere is a little different. Like, sure, there's a few iron cofactors, and maybe it goes a little further than this diagram shows, but for the most part, biology and biochemistry uses a very small subset of elements. Carbon, oxygen, hydrogen, nitrogen, phosphorus, maybe a little bit of sulfur. That takes up the majority of biochemistry. But biochemistry is still incredibly complex. I think it'd be very easy to sit here and say that we and anything else living is a very complex thing. So those kind of properties, whether they're chirality or other things, those kind of predict and say what biology can be. Maybe you can argue that that's limiting, right? Why is biology so limited? Why isn't biology up here? Well, maybe those properties are what limits it. Maybe biology can only have so much chirality, can only have so much molecular weight, can only be distributed across these certain elements. But I like to think of it as a predictive property. Can we predict what future chemical exploration will look like using these particular properties? And what can these chemical properties tell us about life elsewhere? If we see something that looks like this, I would argue that that would be pretty close to life. I argue that would be something that we'd be able to see and say, that is alive and that is not, based on these universal chemical properties. This is something that metabolism and genetics, while they are very informative and really help to distinguish life on Earth, I think the universality is not as strong as the chemical properties that we see. I'll leave it there. I'm happy to discuss this in person during the conference. My Twitter handle and email are down below. Obviously, if you have any questions, please let me know. I'm happy to talk to everyone. And again, thank you for coming.